All right, guys, we are going to continue on with our cellular respiration benchmark notes. And the first place that we are going to continue to is to the aerobic pathway. And by aerobic, we're talking about oxygen being present. This arrow is going to refer to something being with oxygen. All right, our pyruvate, and our pyruvate came from glycolysis. That's what we ended with in our last step, or our first step of cellular respiration, but where we just ended was that we ended with pyruvate. It's going to enter into a transition stage, move on to the Krebs cycle, and then to the electron transport chain in the aerobic pathway. So there are going to be 3 extra steps into the aerobic pathway. This is going to provide almost 20 times more ATP. Actually, what it's going to equal is 38 ATP, actually, on the aerobic pathway. It's going to take place in the mitochondria. Remember, mighty mitochondria is responsible for turning your food into energy. So if we're going to be getting 38 ATP, that's going to be taking place in that mitochondria. The transition stage is going to be the first thing that's going to happen. And as it says, you've got 2 molecules of pyruvate, right? So you've got those 3 carbons. Right? They're going to be modified into something called acetyl-CoA, which is 2 carbons. So what you're going to end up with are 2 carbons, and they're actually going to have this molecule called coenzyme A attached to them in the end. There are a few steps in between there that happen. So what's going in is the pyruvate, and what's coming out is going to be these 2 temporary energy. Remember, NADH is that temporary energy molecule. 2 CO2 and 2 acetyl-CoAs. The location of where it's taking place, this is taking place in the mitochondria as well. Remember, mitochondria is going to be responsible for aerobic cellular respiration. This is going to be a nice little diagram that depicts it. So we've got our pyruvates. First thing that's going to happen is that we are going to lose 1 carbon as CO2. So we've got 2 carbons. That's called an acetyl group. That acetyl group is then going to lose a hydrogen. So let's say that there was a hydrogen right there. All right? Remember, I'm not drawing out all the hydrogens, all the oxygens. But we're going to have NAD come and steal away that hydrogen to make a charged temporary energy molecule. But when that hydrogen gets stolen away, it leaves a blank spot, and the blank spot is going to need to be filled. And what's going to fill it is something called CoA, and CoA stands for coenzyme A. All right? So what you're going to end up with is acetyl-CoA. Coenzyme A is a transport molecule. Its job is going to be to transport the acetyl group onto the next stage. Now, remember, in the end of glycolysis, we had 2 pyruvates, which means this whole thing happens twice. So in the end, you're going to have 2 acetyl-CoAs. You're going to have gotten 2 carbon dioxides, and you're going to have gotten 2 NADHs. Because it happens once for every pyruvate, and 2 pyruvates come out of glycolysis. Then we're going to move on to the Krebs cycle. So now we've ended with 2 acetyl-CoAs. Those acetyl-CoAs are going to move on. Remember, CoA is bringing the acetyl on to the next step, which is the Krebs cycle. So what's going in are the acetyl-CoAs and something called a 4-carbon compound. Cycle. It implies it's going round and round. Or you guys like to tell me that what you end with is what you start with. All right, so that 4-carbon compound is going to be something that got recycled from the last time that the Krebs cycle went around. What's coming out? 2 ATP, 6 NADH, 2 FADH2s. All right? Both of these molecules are temporary energy. That's a new one. All right? FADH2, that's one that we haven't seen before. But again, Still a temporary energy molecule. Only question is, which one is better? All right? Which one's going to give us more bang for our buck? And we're going to find out about that later. Location is that it's taking place in the mitochondria. And the Krebs cycle is going to happen twice. Because if you remember, we ended with 2 acetyl-CoAs in the transition stage, right? 2 pyruvates went in, 2 acetyl-CoAs came out. So now we have 2 acetyl-CoAs that are going to go into the Krebs cycle. 1 acetyl-CoA is going to go in and go around once, and then the next one is going to come in and go around once. So Krebs cycle happens twice. All right, we've got an animation that kind of shows it, and this is a diagram of our Krebs cycle. So the first thing we're going to talk about is that it happens twice. Oops, once for each acetyl-CoA. So, all right, what we're going to have is that acetyl-CoA is going to come in. CoA is going to drop it off. So now we've got 2 carbons, which is our acetyl. All right, that acetyl, all right, those 2 carbons are going to join 1, 2, 3, 4 carbons, and you're going to have 6 carbons. All right, our 6 carbons are going around. It's a cycle. All right, first thing that's going to happen, all right, I went through and I kind of drew out some hydrogens for you guys, gave you guys some ideas about what to look at. I'm just picking some places to put hydrogens. I'm not being actual about it because we're not worried about the actual structure of all of these molecules. We're not worried about that much biochemistry at this point. So we've got a hydrogen here and hydrogen here, and let's say there's a hydrogen there. All right. First thing that's coming in is that NAD is coming in and stealing away 1 hydrogen. CO2 is also going to be taking one of those carbons, which means we're going to have 1, 2, 3, 4, 5 carbons. We're going to have lost that hydrogen that was there. So we're only going to have our 4 hydrogens at the end. Next thing that's going to happen is that another NAD is going to come in, pick up another hydrogen, so another one is gone. CO2 is going to take another one of these carbons, which is going to leave us with 1, 2, 3, 4 carbons, and we're now only going to have 1, 2, 3 hydrogens. We got rid of that one, all right? Now, we also have a phosphate on there. All right, I'm not really worried so much about phosphate, whatever. All right, our ADP is going to come in, take that phosphate, and we're going to get an ATP. All right, which is now going to leave us with our 4 carbons, with our 3 hydrogens, and that's it. FAD is now going to come in and pick up 2 hydrogens, which is going to leave us with 4 carbons still, because we haven't lost any carbons, but now we've only got 1 hydrogen. We lost 2. That same molecule, continuing on, but this time what's going to happen is that NAD is going to come, pick up that hydrogen, and we're right back to exactly where we started. The 4 carbon that's going to be recycled through for the next acetyl because it happens twice.